All right, so the next part of the session are going to be 3 contributed talks. So I first wanted to recognize we got a large number of really excellent poster contributions for this meeting. So I would encourage everyone to really, take some time, look at the posters during the session. They're out there now. So anytime you get a chance, look at the posters. The meeting organizers had a really hard time selecting among these really excellent contributions a small number of poster abstracts to select for these contributed talks. But I'm really happy to present the first session of 3 contributed talks. And our first speaker is going to be Alex Brown from National Jewish Health.
Thanks for inviting me to come here. So I'm from National Jewish Health. I'm an immunology PhD candidate at the University of Colorado. And our overall research question here is to understand how does the selecting MHC allele affect the TCR repertoire? And to approach this goal, we want to understand what controls the repertoire of T cells on naive T cells in mice. And we think that this might help explain MHC allelic influence in autoimmunity. So there's a technology limitation here. So methods just don't really exist yet to pair TCR alpha chains and beta chains on all naive T cells in a mouse, especially when we started this project. So to circumvent this problem, we decided to fix the beta chain. And we're just looking at the TCR alpha sequences here. So I just wanted to give you that as a primer. But the resulting mice that we have a fixed beta chain and only one functional TCR alpha locus. And then we're looking at these mice that are all black6 background, and we're looking at them that have different class 2 haplotypes. So I'm just showing you the mice here. They're BB, FF, G7, G7, or SS. So these are MHC homozygous mice, and we're looking at the TCR alpha chains that are expressed by naive CD4 T cells. And just to show you first, so we can see that the BB animals, for instance, if you look at their TCR repertoires compared to other BB animals in our cohort, their repertoires are extremely similar to each other. And there's virtually no overlap between the mice that have different MHC class 2 molecules as far as their CD4 selected TCR alpha repertoires. We can do other kinds of comparisons that account for the number of particular clonotypes that appear. And we see that these similarities are very consistent. So if you look at all the other comparisons that we have, the phenomenon is the same, that these repertoires are effectively virtually non-overlapping depending on the particular haplotype that they're selected upon. So we want to know what happens if we cross these mice and introduce 2 MHC alleles, making them MHC heterozygous. So just looking back at our MHC homozygous animals, this is what they are. And we've crossed them various different times to create MHC heterozygous animals. Then they inherit a class 2 molecule from either parent. So I'm just going through and showing you the different MHC heterozygous animals. And then we looked at the TCR alpha repertoires to see how do those repertoires differ, what do they share, et cetera. So using these same kinds of comparisons, we can see that, for instance, the B cross F animals up here, they look similar to other B cross F animals as far as their TCR alpha repertoire is inherited and somewhat consistent. And they also share sequences from either parent, which should be maybe not very surprising. But we see this consistently in all the different crosses that we have. And just to hit home this point, so the FF TCR repertoire is inherited or effectively selected by particular MHC class 2 molecules from the parents. And those repertoires, the beige colored and the blue colored, are virtually non-overlapping. And the B cross F animals then inherit those same class 2 molecules and should then elicit the same sets of TCR repertoires from either parent. But they should be able to elicit the sum of the 2 parents as far as the repertoires are concerned. And then they can also make these mixed molecules, which should be able to elicit unique peptides that are presented to the surface in thymocytes. And this should elicit unique TCR clonotypes that only show up in the F1 mice. So this is our expected TCR repertoire. It should be much larger than either of the parents. And what we actually see is the actual repertoire of these F1 animals is heavily restricted, which was very surprising to us. So these circles here are just a representation of the number of unique clonotypes that we see in the composite repertoires from 6 of these animals that we've sequenced. So for instance, the B circle, that is the composite repertoire of 6 mice from our cohort, and we're just looking at all the clonotypes that show up in those 6 mice. And you can hopefully appreciate that that green circle is much smaller than either of the parents, and we would expect that that green circle should actually be able to composite all of the sequences that are shown in those weighted Venn diagram plots. It's also probably worth mentioning that we see the same phenomena in different crosses. I'm just not showing you that for the sake of time here, and there's other ways we can make these comparisons, but this is just visually an easy way to show this for a short talk. So we want to know, why are some of these sequences missing? So some of these sequences are definitely missing in the MHC heterozygous mice. They never show up, and they always show up in the other parents. So if you look at TCR development, there are many different checkpoints and ways that we think that these sequences could go missing in TCR development. So I'm just going to focus in on the stage of a T cell from going from a double positive thymocyte to selecting to commit to the CD4 lineage. And we think that these TCRs are deleted by clonal deletion from class 2 molecules that are presented from the other parent and or the mixed molecules. It's worth mentioning that we have done a lot of different studies to see in different checkpoints of where these sequences could go missing. I'm not really going to get into that today, and I'm just going to focus on this first step because it seems like this is actually what's going on. To test this, we've made very unique mice. These mice allow us to look at the class 2 molecules that are expressed from either parent by creating tetraparental mice. So these tetraparental mice are formed by the fusion of 2 fertilized embryos at very early stages in development. And what you get is these striped mice. So these striped mice effectively have just the class 2 molecules from the 2 parents. And we can use these as counterpart mice to compare to the F1 animals. And the F1 animals, in comparison, are able to express the class 2 molecules of both the parents and these mixed molecules. So what we see is that these tetraparental mice have much larger repertoires than either the 2 parents or these F1 animals in particular. So this shows that these tetraparental animals are able to make these TCRs that are missing in the F1s, and the F1s are definitely missing these sequences, and they have holes in their repertoires. Just going back, this is one of my last slides. So bringing this back to our overall research question, there's virtually no overlap between the TCR alpha repertoires of the mice that have different MHC haplotypes. The F1 animals, they share some sequences from their parents. However, they develop very large holes in their repertoires. And we think that this is indicative of an inverse relationship between the number of MHC class 2 molecules that can be expressed and the size of the TCR alpha repertoire. There's my last slide. So what are the consequences of MHC heterozygosity? So we want to know, how does this play a role in infection effectively? So the peptide repertoire is expanded, allowing you to survey more antigens. What is the consequence of this? And we think perhaps that you're preventing certain diseases by eliminating unwanted autoreactive clones. And we've done some analysis to do this, so I don't have time to talk about this, but we've developed large screening assays to clone and test over 900 clonotypes from our TCR alpha repertoire data. And I've identified many of these clonotypes are actually missing because they are autoreactive. So thanks for listening. I'll keep it short. So come to my poster if you want to learn more. Thanks.
Oops. Nice study. I might have missed this. When do you sample the T cells before sequencing? At which developmental stage?
So we're looking in the periphery. So we're looking at naive CD4 T cells.
Okay, is there any possibility that you could sample double positive cells in the thymus, so before negative selection?
Yeah, we also have that data.
And what does it say?
So the double positive thymocytes, they effectively don't look like the peripheral repertoires as you might expect, but the double positive thymocytes from different MHC haplotypes are also different from one another to some extent. But once you go into the periphery, they're wildly restricted to particular haplotypes. Okay, so negative selection does contribute to creating those holes that you observe in the periphery. Yes, yeah, so. Okay, thanks.
So how did you introduce your TCR beta rearrangement? So was it a knock-in? Was it a transgenic?
Yeah, they're TCR beta transgenics.
Okay, and so in your setup, so all your cells express the transgene. Did you check about that?
Yeah. They all express that particular TCR beta. They don't express any other possible TCR betas.
Okay.
And we've also checked that they only have one possible functional alpha locus to rearrange.
Okay. And in terms of differentiation from double negative to double positives, because the beta chain is pre-selected at the beta selection, so before double positive. So is there any difference depending on the MHC, since we still don't really know how things are going at that stage?
It doesn't seem like there's
Okay.
much difference there. So we also want to look at this in an unrestricted setting too, because there could be possible limitations to just having this fixed beta chain. We want to see, does this also occur in an unrestricted setting? In our preliminary analysis of that says that is also the case.
Okay, thanks.